Thank you, Thomas. Good morning, good afternoon, actually, everyone. So my talk today will be kind of complementary to what we have just heard, since I will basically focus on large-scale network interactions, but in ongoing brain activity, so of intrinsic activity. And I will do it mainly from the point of view of MEG, but also relating the MEG power and results to some fMRI knowledge in that field. So in doing this, I will try to convey you some messages. And the first one is something that has already touched upon by other speakers today, which concerns the richness of the electrophysiological in general and MEG in particular data, and how this can be related to extract the important information in intrinsic connectivity. Then I will talk about the different methods that this kind of data, MEG data, can offer to approach the problem and to look into different spatial and temporal details to this problem. And then finally, I will relate the MEG findings to the well-known fMRI findings in this respect. So why to focus on ongoing brain activity? The reason for that is kind of well-known today, because system neuroscience has moved from considering ongoing brain activity as from an idling state to a real substrate which the brain has and which can be shaped by, for instance, experience. So in this framework, it is well known that fMRI has done a great job in identifying what have been called so far as resting state networks, which are basically patterns of ongoing brain activity, which have been identified starting from 1995 from Barak Biswal, which has already been named today. And later on, these like spatial temporal structures have been deeply studied, and with the, do we have the laser pointer? No.
The mouse.
The mouse, okay, thank you. With the idea that if you like seed, so you choose one region in the brain, then you can highlight structure which are correlated at rest, so when the brain is resting, and the red structures share the same spatiotemporal pattern with our seed, while there are other regions which behave out of phase in respect to that. Okay, this initial notion has been largely extended, and basically in the recent years, full coverage of the cortex has been obtained, which like parcels somehow the cortex into different networks with different functions. So what has done MEG in respect to all of this? MEG has contributed along these lines starting from like 2005 or so, revealing that there are slow fluctuations in the MEG signals, the coupling of which moves in the order of seconds, can resemble to some extent the results that have been observed with the fMRI. Specifically, this is one piece of work, one of the first ones, in which you can see the comparison of fMRI and MEG, and this is the first kind of effort that has been made in the community, so to show what is the resemblance of the two patterns. And along this line, other groups, like in the UK, have shown that there is some resemblance, for instance, between the fMRI motor network and the MEG network in the beta band with different approaches, which we will go more in detail later during this talk. So what is like this? I mentioned this idea of slow fluctuation, and so just to be everything on the same floor, I want to highlight what I mean by this. So if you have the MEG signal, which is here represented by the blue signal over this, and you have it in one region and in another region, then what you are looking for is this red signal here, which represents what is called the amplitude envelope of the signal. And then you look for the coupling of this red curve here with another red curve over here. The idea, which is related to this concept of amplitude envelope correlation, is that bold, fMRI bold, and amplitude envelope of unlimited power in MEG or EEG are both related to power consumption. So in some way, you can extract the fluctuation of power from the MEG signal, which is this slow varying feature over here, and look at the correlation of this information in the brain in a very similar way to what is fMRI correlation analysis. And okay, I mentioned correlation, but of course you can do it with correlation, and you can also estimate the coupling by using other methods, such as partial correlation or independent component analysis, as we have already heard about today. All these methods are in the time domain, are data-driven, meaning that I'm not talking about method which required the assumption of a model, as many of our speakers have done today. But I will just go basically into this simplest framework of analyzing correlation-like quantities between amplitude envelope. But as we also have heard today, we are not aiming at doing this at the sensor level because of the very many issues which affect the EEG signals, but also the MEG signal as well. So our first goal will be that of deriving the brain signals from the MEG signals measured at the level of the helmet of the sensors. For doing this, we have already seen that we need some kind of inverse approach, an inverse method, which will give us the activity at the source level, what is usually called the source level. And then we can estimate connectivity of this amplitude envelope according to different metrics. But the big but of this story, which you have also heard today, is that we have to cope with this volume conduction issue, which usually in MEG is also referred to as source leakage, because we are not analyzing our coupling at the level of sensors. Rather, we are going into the source space and try to assess the relationship at that level. Nevertheless, what happens is that we are not getting rid of this issue if you just localize the sources. And the reason for that is that if you consider very close by voxels or brain regions, then they are assigned with kind of very similar activity. And this depends on the point spread function or blurring characteristic of the specific inverse method that you are using. So you have something which is connected, like fakely connected by itself, and the extent of this depends on the inverse that you are using. So one possible option for that is to get rid to mitigate this effect in a similar way of what we have been hearing about with regard to the sensor level, and is to try to remove some synchronous activity from one source to the other. Now, in the framework of amplitude correlation, what is usually done is what is called signal orthogonalization. And actually, let's consider two sources here. Which one of these is the test source, and another one is, for instance, our seed source, so our reference point in the brain with respect to which we want to calculate connectivity. Then what we can do is to design some kind of general linear model in this way, in which we obtain the orthogonalized source 1, so this one, from which we have, which is obtained as the first source itself minus a given coefficient times the second source. So this means that with this GLM model, we will remove from the first source all the synchronous contribution from the second one. And this will give us some orthogonalized signal with respect to the seed. This is just one option of, the equation that you are seeing is just one option of achieving these orthogonalizations, but an extensive review of this can be found in the O'Neill paper in 2015. But anyway, the basic idea is that still you remove some synchronous activity that is shared between the two sources. And this is typically done in these amplitude envelope studies. And so if you do this kind of procedure and then you estimate your amplitude envelope and then you do some correlation of that, you end up with kind of nice pictures, such as those ones which are shown in the paper of your KIP in 2012, in which you can see some MEG coupling of the auditory, somatosensory, and visual or primary networks in the resting state in specific frequency bands. So just to wrap up, say that you want to go with this path and you use your MEG data in order to analyze slow fluctuations and to see how they relate to fMRI connectivity. Then what you should do, of course, you acquire your resting state data at the sensor level, and then you choose some source estimation approach. which of course gives you some spatial topography plus a time course. And then you need to cope with this source leakage effect. So one way of doing it is to go for this signal orthogonalization, and for instance, with that approach. And then you can either try to obtain directly the amplitude envelope, or want to extract some specific feature of your signal, such as a given frequency content. So you could bandpass filter your data, for instance, in the alpha frequency band, and then estimate the amplitude envelope of the bandpass filtered, or do directly amplitude envelope estimation of the whole signal with the full frequency content. But anyway, then you end up with these slow fluctuating signals, and you want to do some kind of correlation or coupling within them. Of course, Pearson is the easiest way to go, but you could really apply also ICA directly on this amplitude envelope and see what the patterns of connectivity look like. Okay, and finally, you end up having the kind of maps that we have already seen, which are usually called in the MEG domain amplitude envelope correlation maps. But this is not the whole story of what you can get from MEG. It was nice to see, like in the 2010 and immediately following years, that there was a kind of resemblance, because this made everyone confident that the fMRI results had some electrophysiological correlates, also by using non-invasive approaches. But as we have already seen, the MEG signal has a much higher temporal resolution, and which has not really been exploited in this approach. So we ask ourselves if we can exploit the full temporal information that is contained in the MEG signal, and at the same time find some pattern which still have relationship to this idea and concept, the notion of the resting state networks. What I will talk about is phase coherence, considering it as a possible tool for investigating coupling between neuronal populations. And specifically, there is actually some physiological reason for wanting to use this kind of measures to map our connectivity. And the physiological reason is deeply related to the concept of communication through coherence, according to which if you have two neuronal pools, one presynaptic pool and one postsynaptic pool, you have the higher chance of communication between them only if they oscillate in a way such that the respective neurons fire at the crests of these two oscillations, and this firing is either synchronous or with some time lags. Actually, there is a bulk of evidence also from invasive electrophysiology that there are lags in the brain, and this makes us confident that maybe we don't see the whole story, but still we can hope to see something out of this story. A lot of work has been done from people working in methods to try to develop different measures which are more or less all devoted to this specific aim, so that of studying phase coupling between two groups of neurons. And this is a long list, which is, despite being long, not fully exhaustive. So since, of course, we cannot cover all of this, I will focus mainly on the frequency domain measures and, again, data-driven approach. And even more specifically, I will focus on linear functional connectivity metrics. But if you are interested in some nonlinear approach, I will have a symposium on Thursday morning in which I will go in more detail of the nonlinear phase coupling approaches. Here, also for the sake of the educational content of this meeting, I will go into this approach, which is very simple because it assumes that you have two sources and they both oscillate at the same frequency. And so it resembles pretty much the picture that we have been seeing before. And we ask ourselves how we can assess the relationship between these two oscillations. So in order to understand a little bit this family of methods, then we need to go a bit back into signal processing and to define some quantities. So let's consider just one of those oscillations. So this is one signal oscillating in time and with some characteristics, one of which is the amplitude, which is this value A, which varies in time, and another information is the phase, which gives you like the relative position of one point with respect to the 0 of your axis. This kind of information can be nicely represented in the frequency domain. So other than just showing this story as it is plotted now on the screen, you can view the same story from different perspectives. So you can draw this stuff as this signal as a vector, the amplitude of which is the amplitude of the signal, and the angle, okay, here it is, and the angle which it forms with the x-axis represents the phase of the signal itself. And if you want to put some maths in it, you can write this vector in this way. So this is the amplitude, and this gives you some relationship of the quantity with the angle. But now, what we are really interested in is not just one signal and these amplitude and phase characteristics, rather the coupling between two signals. So we have the same story. We represent the one signal in this way, and the other signal in this other way with this mathematical expression. And then we look for synchronous or like lagged coupling in between the two. And if we do this, this means that we are asking ourselves whether there is some phase difference between these two vectors, which is represented here by this angle, and whether we are able to detect this phase difference, which will be like the marker for phase coupling between the two. And from some math, the building block of this story would be now the product of the amplitude of these two vectors multiplied by the exponential of I times, this time, the phase difference between the two and not the phase itself. Okay. So starting from this point of view, what does it mean that two sources are coupled according to some phase coupling, or they are not? Actually, what we are looking for is this kind of situation. Consider that you have one epoch of your data, which, since we are considering resting state activity, is not locked to any specific event. And in this epoch, your vector representing the coupling between the two signals is this one here. And then you consider the second epoch in which you have something similar, and so on and so far. You do it for all the epochs in which you have chunked your signal, and then in the end you average. So if you are able to find a vector with some length, meaningful length, and with some angle with respect to the x-axis, then this means that we call this situation a situation of asynchrony between the two sources. Of course, we are talking about lagged synchrony, but still synchrony. While the situation could be just opposite. So suppose that you do the same kind of analysis, and this time you end up with estimating vectors which vary in amplitude, but also in the angle that they form with respect to the x-axis. So overall, when you average along the duration of your data recording, you will end up with possibly a very small signal with a kind of undetermined phase, and then you cannot really use this kind of information to derive anything about the coupling of your signal. So we usually refer to this situation as a situation of low synchrony. Okay, but this quantity that I've been highlighting over there, which is like the mathematical definition of the coupling within one of these epochs, now needs to be extended to take into account this procedure of averaging that we have been doing along our data duration. And if we do that, then we end up with the definition of the most common measures for phase coupling and connectivity metrics. Actually, this is what is usually called the complex coherence. So it's a complex value, which is a normalized value, and the importance of normalization, which were mentioned before, still holds also for this kind of measures. And I mean, from this building block, then you can extract coherence magnitude, then you can extract the phase locking value, the imaginary part of coherence, or the imaginary phase locking value, or many more quantities that are different in, for instance, the normalization factors, or some slight differences in there. But all of them come from this idea of synchronicity that I've been depicting with the previous picture. Okay, so we are happy. We use our MEG signal with its high temporal resolution, not the amplitude envelope. And we use this kind of approach which directly tell us if two sources are phase coupled or not. But still, of course, we have the problem of source leakage and field spread, which is here solved in a very similar way to what was done before for the amplitude correlation. So the idea is, again, to throw away what is synchronous. But likely, since I've done part of my PhD with Guido Nolte, I know how to say it properly, and the proper way of depicting this story is that if you have a non-vanishing imaginary part of the coherency, then this means that this can only result from phase-shifted interaction. So it means that if you have your vectors, which represent the synchronicity of the phases between the two vectors, and you consider just its projection along one of the axes, and that's basically the idea of imaginary coherence, this will be non-zero only if there is some phase-shifted interaction, actual phase-shifted interaction in the data. Of course, the other side of the story is that if you have perfectly synchronous coupling and just that, you will not be able to detect it with this kind of approaches. But if you have evidence, as we have for lagged coupling in the brain, then this might be one of the ways to go. Okay, now again, to wrap up this part of the story, if we now want to use our MEG data to do the other pipeline, and so to explore the intrinsic features of the high temporal structures of MEG data, then we still have to choose some inverse method. And then what we can do is to go directly, for instance, into the frequency domain. And this will give us like a set of oscillation, which weighted will like give us the full signal that we started with. We can represent this information in the way we have just done. And then we can use some measures which are not immune, rather robust in the sense that I've just been mentioning, to source leakage, and obtain some lagged coupling in the brain, such as this that I'm showing here just for the purpose of having some visualization. I will go in more detail later on that. And so in this case, we will have obtained what we call phase coherence-based maps. But something that I've been completely ignoring in this part of the talk is that actually when you localize the sources of MEG activity, usually what you end up with is a quantity which is not only represented by one number, as for fMRI. So for fMRI, you have one brain area, region, voxel, whatever, and you have a signal which can be assigned to that area. Well, if you do the same for MEG, then you usually choose some reference system in the brain and you project your data from the sensor level onto that reference system, which lives in the brain space. And if you do that, you end up with having more than just one signal. You usually have two signals, which basically go together, but still the full information of MEG data is contained in both of the signals. So if you want to fully exploit this information that can be derived from MEG data, other than just throwing away one of that and concentrating, for instance, on the one which has the maximum power, you can go into some multidimensional or still multivariate approaches, which take into account the contribution from all signal components. Okay, one of those examples is the multivariate interaction measure, the results of which I will show to you in a while. But the idea is similar to what Laura was mentioning this morning when you go to the bivariate case to the multivariate case. So you know that the information is contained in more than two things, and we want to take into account for the full amount of information. The way the problem is solved is different, but the basic idea is the same. So we can use this kind of multivariate or multidimensional phase coherence approach, which from a theoretical point of view share the same idea of the imaginary coherence itself. and try to map our networks in the brain with this kind of approach. That's actually what I did some years ago, starting from the dorsal attention network, which has been well studied in fMRI and is characterized by rather superficial nodes. So in some sense, it's really ideal to be looked at with MEG. And actually, I will not go into the details of this, but these are the measure nodes of the network. And if you see, for instance, 2 of them, then you could do some maps, frequency-specific maps of the activity relationship between these brain regions and the rest of the brain itself, and then do some conservative conjunction procedure in order to highlight the patterns that are common to both these nodes of the network. And actually, we did this with some success, because our findings showed that we had both coupling within the dorsal attention network and between the dorsal attention network and other resting state networks. For the first part of the story, which is the coupling within the dorsal attention network, actually this slide wants to highlight the delta band and alpha band features, which kind of resemble, at least contralaterally to the seeds that we have chosen, the classical patterns of the fMRI dorsal attention network. So we were very happy with these results, showing that not only amplitude envelope correlation, which is in a way similar to fMRI signal, because we have made it to be similar to the fMRI signal, but also phase coherence, which keeps the high frequency somehow content in the signal, resembles some patterns and topographies in selected and specific frequency bands, which are looking pretty much like the resting state networks as seen in fMRI. Moreover, as I said, that's not the whole story, because we were also able to extract some features from our data which speak about a coupling between the dorsal attention network and other primary networks, such as the motor network here in the specific beta frequency band, and the visual network here in the alpha frequency band. And so taken together, these results somehow speak for a frequency-specific coupling between the dorsal attention network and primary visual networks, which is observed in the resting state, but still involves the frequencies which are known to be involved during visual motor attention tasks. So somehow this experience and learning works also for the frequency content that we have been able to extract in our signals. Okay, last but not least, we have heard this morning that we are not only interested in coupling. This is functional connectivity coupling-based measure, but we are also interested in directionality. And I will not go into the details of this because it's really kind of brand new on the one hand, and also requires a lot of maths here. But we have a poster on this like on Tuesday, and it is possible to derive some phase-based measures, which are also, again, multivariate in nature, so take into account this higher level dimensionality of the data, which we have already just seen, and exploit, again, from the frequency domain point of view, the idea that has been already brought about several times this morning, which is that temporal precedence indicates somehow the leading and the following sources in the brain. So the idea is very similar to that, but all the details are derived in the frequency domain. But I wanted to have this last slide here because this speaks again for some resembling of resting state networks and as seen by fMRI, and resting state networks as seen from MEG data. These are the MEG data of the Human Connectome project, in which part of the methods that are being used in this talk have been also developed. And what we see here are the major nodes of the more well-known resting state networks, which are represented on the cortex as colored dots. And they are superimposed onto our MEG results, which this time indicates some directional coupling. So this means that we have seeded the visual cortex, and everything which appears red in this slide is something that leads the visual cortex. So it's from that region to the visual cortex, while everything that is highlighted in blue receives some inputs from the visual cortex. And so the only thing, I will not discuss the results. Again, if you want, I will be happy to welcome you at the poster. But the idea here is just if we consider the dorsal attention network that we have just been mentioning in the previous study, we can see that, for instance, the parietal part of the dorsal attention network is leading the visual seed, so the visual network, in the alpha frequency band, which is actually the same frequency band in which we observed the functional coupling in the other study and the other data, highlighting that this time we do not recover with directed measure the full pattern of the network. We don't see the frontal part, but still we see a lot of information that comes from MEG phase coupling in this respect. Okay. So in conclusion, I hope that we all are convinced that MEG data have a quite large content, meaning that they have exquisite time resolution, which can be explored as it is or smeared somehow to extract something that goes closer to the bold signal, and the frequency information which can be used in order to disentangle different contributions in this framework, and that there are different and very many methods and approaches to study functional connectivity with MEG, and they are somehow complementary, at least for what regards the amplitude envelope correlation on the one hand and the phase coupling to the other hand. And for their nature of these methods, then they share different relationships to the fMRI functional connectivity as seen by resting state networks. And finally, my understanding of all this story, as seen from MEG, is that MEG is able to identify some frequency-specific system which possibly are part of resting state networks. So they identify subsystems which belong to the larger concept of resting state networks as it has been brought about by the fMRI community. Okay, so I thank you for all your attention. I thank Laura and Thomas, of course, for organizing the great symposium, the people at my university, especially Vittorio and Alessio, and the people at WashU who collaborated on this work. Thank you for your attention, of course.
Okay, thank you very much. Are there questions from the audience? Everybody's in for it. Yes, please.
Wow.
Great talk, thank you.
Thank you.
I'm curious if you were to look at other frequency bands. I missed the first 5 minutes, so maybe you talked
Okay,
about
don't worry.
this, but you know, if you were to examine other frequency bands, if you go back to that slide where you had the distribution.
This one? No,
no,
the previous
earlier
ones.
with alpha and delta.
Yes.
Yeah. I mean, those aren't, to my eye, those aren't that distinct from one another between the delta and the alpha. And if you were to, you know, examine perhaps at a lower threshold theta range or gamma range or beta range or something like that, you know, and just correlate.
The topographies, you mean, or something?
Yeah, like a Pearson, just the overall similarities across those different distributions, you know, barring statistical significance and thresholding and maybe clustering. I don't know if you do some sort of cluster thresholding here, but you know how similar they really are across frequencies.
No, actually, we didn't measure that. What I can tell you is that for the beta, actually, we don't have anything within the network. So the beta is this one. And also the extent, let me check if I'm saying, yes. So like if you look at the amplitudes, so the amplitudes for the alpha and for the beta are more or less the same. So it's not just a matter of lowering the threshold and you will see the alpha pattern in the beta, that I'm sure. For the gamma, we didn't see anything, so I have to really to say it, to admit. We also explicitly wrote it in the paper that there was no specific topography which was surviving our statistics. While for the theta, actually it was more or less the same case, but what I remember is that if you look to some kind of not really thresholded data, but if you lower your threshold just for fun and take a look, then the topographies were more similar to the beta than to the delta and alpha. So it looks like if there is, and we also have one graph in the paper in which we show that it's really a double-peaked story, because if we like take these two regions here and plot their connectivity with respect to frequency, then you will see that they have a maximum in delta, another maximum in alpha, but not in theta and not in the other frequency band. But still, no, meaning that we had explicitly correlated the patterns also to the fMRI or so. We didn't do that. That could be a nice thing to do also with the human connectome data, in which we have fMRI and MEG on the same subjects, and that could be easier.
There's another point to make on what you said, and to open up this discussion a bit. When you have simultaneous recordings of now EEG, but we don't care much about that at the moment, and fMRI, and you do just the spectral correlates of the resting state networks. You use the resting state network fluctuations as a regressor for the EEG power spectrum. You see that the spatial distribution of these correlates varies by frequency. And that just tells you that the different subsystems of these resting state networks must have different frequency distributions, and that
some signatures, some frequency-specific
and that
signatures.
makes these resting state networks something quite complicated in terms of of how they interact in terms of oscillations or frequencies, and that's a puzzle that we still have to solve.
Yeah, I agree on that. And also I skipped, I mean, I briefly mentioned, but other speakers today will address also the issue of how cross-frequency clubbing is related to this story, which is another part of the business. Okay.
But I think it's really interesting that we come to the point that we should not just try to map the electrophysiological data to the fMRI data.
No, I agree on
it's
that.
way more complicated. And
I agree on that.
You talk about subsystems and all this, and I think this is really the way to go.
Yeah, okay. Thank you.
Okay, we have 20 minutes coffee break, and we start at 10 to 3.